So I'm going to talk about some of the imaging science research we have going on at CDRH. And I'm in from the Office of Science and Engineering Labs, and I'm the acting director for the Division of Imaging, Diagnostics and Software Reliability. So I'm sorry, we don't have a ton of research directly related to radiation oncology, but most of our work's related to radiology and imaging. So I'll give a quick overview. What I'll stick with is probably this first part, sort of programmatic areas that we have for the research areas. I have a couple of specific projects. I work in computer-aided diagnosis and machine learning, as well as quantitative imaging. We also have this other interesting project on in silico clinical trials. So how would you develop in silico models for the patient, for the imaging system, as well as for the observer, the reader aspect of that. And so we're now trying to make process that streamline clinical evaluation, where we could try to reduce the need for some of the clinical trial types of things that we do in some of the systems we have that come in. So I'll just sort of give you a brief overview. You might ask, why do we even have a research group in FDA? What we try to do is support pre-market device evaluation and post-market quality control and surveillance. And what our real role is to expand regulatory science to better promote public health. We do this through test method development. We look in our office at things like biological, chemical, material testing, physical testing for devices. What types of testing could we do on an imaging system or a therapy system that would lead us to new standards and evaluation methods? We also do a lot of work in phantom and phantom testing. So we develop phantoms in-house, again, for QA/QC potentially, as well as for potentially pre-market evaluation. One of the big areas we're working on now is in silico modeling of multiple chains in the imaging process. And then we look at things like theoretical predictions and limits and so forth to evaluate how well systems potentially should be performing. The other aspect that we work on a lot in my laboratory is clinical and non-clinical study designs. So we have a lot of expertise in how you set up, especially when you're incorporating readers or clinicians into the process, how you would set up those studies, how you would evaluate them. And then we have a lot of work in the mathematics and the statistical analysis methods and how you're going to evaluate those studies, size those studies, and so forth. So I'll talk about 3 different programmatic areas. We have imaging physics. We have roughly about 7 or so full-time staff members. When we're looking at evaluating image quality for emerging regulatory topics, we've got a lot of work in 3D breast imaging that we're working on. We also look at older modalities, things like CT, when new claims come in. So when we get to things like iterative reconstruction methods, we're interested in things like dose reduction claims. How would we validate those? What type of data or phantom studies would we need? When do we have to go to clinical analysis? And then newer implementations, things like spectral CT and so forth. How would we do material characterization and those types of issues? We also have work in newer modalities, things like elastography and ultrasound. How would we quantify those measurements and so forth? And then how do we come up with physics testing for those? What are the FAMs we could use to validate those particular claims you might make for new systems like that? We also, again, do work on what we call tools for virtual clinical trials. We have a lot of expertise in modeling the physics of imaging, X-ray physics, and interactions in tissue. So we do a lot of work in both the detector side, how an X-ray interacts with the detector, either producing photon arrays or direct interactions, as well as the interactions in patient scatter and so forth. And then one of the other things we do is display characterization. We have lots of methodology in our laboratory, probably one of the best laboratories in the world for characterizing displays and developing methods for display characterization. We're now interested in sort of new types of technology. If we're going to do stereo display, where you're going to look at things in 3D, how is that going to impact the reader, the clinician's ability to look at that data? And as well as new modalities like color transfer. So when looking at endoscopes, or particularly things like digital pathology, which is something that we're trying to move towards on the market, everyone's interested in that. How do you keep the color fidelity, because it's an important part of the clinical decision process? We have a diagnostics and assessment methods laboratory. This is the one that I work in mostly. We have efforts in quantitative imaging. We do a lot of work in phantom development for that and statistical assessment framework. So this, again, ties into radiation oncology as well. When you're looking at imaging and you're looking at imaging studies, what's the variability coming from those studies based on different imaging acquisition or different image acquisition parameters and things like PET? What are the dose differences that are causing variations in what you're seeing? So if you're physically measuring a lesion, how well can you make that measurement? And what can you do practically for the imaging system? What we've done in some of the research has shown that for things like CT, you can actually reproduce, if you do this in a theoretical way, you can really reproduce that lesion by the reconstruction process if you know all the information going in. The problem is you don't know that going in, so you get large variabilities in your ability to make these measurements. Or if you have a clinician coming in using software tools that are semi-automated, you'll start to see a lot of variation in how large or small that lesion is. Now, if you have a very large lesion, maybe that variation isn't so large, so big that it really is an important factor. But when you start to talk about smaller lesions, things that are changing, patient motion, things in the lung and motion artifacts, which I'm sure you guys deal with all the time, you start to see these larger variations. That leads to larger dose profiles for things like therapy, and it leads to more imprecision in whether you're making a medical diagnosis as well. So we do a lot of work in trying to understand how to assess that, what are the factors that are really contributing to that measurement error, and then how can we try to control that error as best you can. We do, again, a lot of work in computer-aided diagnosis and machine learning. I won't go through all of these projects, but we look at things like calibration. We have a reasonable number of devices, not necessarily a lot on the market, but we have a number of devices on the market, but they all give you sort of arbitrary scaled data. So a clinician's going to get a number out of a system, and they're going to have to try to interpret that number without any real context to it. So what we're trying to look at is ways to calibrate those measurements across different vendors, different platforms, to come up with a scale that may be related to probability of malignancy. We see this in things like ultrasound bone density measurements, where you have different vendors with different ways of measuring that, come up with different scales to measurement. So you have this incompatibility, which makes it more complicated for clinicians to figure out how to utilize it or change from one system to another if you buy new hardware. So we have work doing on that. One of the areas that I'm most interested in is this idea of assessment of continuous and adaptive learning algorithms. So we're moving towards more of these computational systems that are going to be implemented in clinical diagnosis, whether it's to aid the clinician as a measurement or to potentially take over and give clinical diagnoses or give feedback directly and make measurements. And one of the areas that we're struggling with is if you're going to have this ability to acquire large amounts of data, large imaging databases over potentially multiple locations, and you can do that in semi-real time, how can you develop a process that you could retrain that algorithm, improve it as it's moving forward? And so this is a real struggle because there's lots of questions about the integrity of the data, how much of a variation you want to see in algorithms and so forth. And so it's one of the areas that we're trying to put some effort into and trying to understand how we can approach that from a regulatory perspective. We also do a lot of work in what I call reader studies, where we're really evaluating a device. It's not the device itself we're interested in. It's the fact that that device is completely integrated into helping the clinician. So when we have a computer-aided diagnosis system, it doesn't work by itself. It has to be how the clinician interprets that data. Same with an imaging system. You present an image to the clinician, you can produce a perfect image, but if the clinician can't interpret that perfect image, it has the perfect information, it's not going to be a useful product and we can't approve it. So we need to understand how we're going to evaluate these devices when we have a clinician in the group. And one of the issues with clinicians is there's variability associated with it. And we've talked to a lot of people, even clinicians, radiologists that we deal with a lot, and they're amazed at how much variation you would see when you do a study and you look at mammograms and how much variation you see. And it turns out, that turns out to be a big source of variability. It doesn't actually mean the device isn't performing well. The device could be very good, but when you have this added variability of a clinician and all this variation, it's hard to differentiate whether that device is working better or the same or worse than a predicate device. So we do a lot of work in trying to define how you do these studies, how you reduce that variability, control it as best you can, so we can really be testing the device itself, because that's really what we're interested in, is the device itself, when integrated with the clinician, going to improve performance. So we have these different approaches called multi-reader, multi-case study designs. We do a lot of work in how you would size those studies, how you would analyze those studies. We're also looking at things like different endpoints. What's the right endpoint for a clinical study when you're talking about imaging devices and so forth. Things like utility analysis. What's the utility of that particular device and how do we measure that? And then we have this new area in digital pathology where we're doing a lot of work in trying to understand. It's an interesting scenario because you're looking at optical microscope, which is the way pathology is done on a general basis now, still in the US. Not necessarily true in Europe, but in the US. And we want to convert, or people want to convert that into digital pathology where you're going to look at it on a digitized screen. And just to have the clinician look at exactly the same location is a problem. So you can be looking at a slide through an optical microscope, make a diagnosis, and if you look at it digitally, you may have a large variation because you actually are looking at a different area. So we're developing methods where we can control this, we can make sure they're looking at exactly the same feature, and again, we can evaluate the technology, the digital pathology technology, and not get confounded by the clinical variation you might see in a diagnosis or a measurement. And then the final area that we have a program in is software reliability, and this is again a large area. It's a complicated area, and it's trying to do assured high confidence cyber physical medical device software systems and validation. And we end up with, some products with embedded code, maybe just hundreds of lines of code. But in radiation oncology, you have millions of lines of code of radiation planning tools and so forth. There are clearly going to be inherent bugs within that framework. And the question is, how can we build quality systems? How can we help vendors build quality systems and give us the right level of information that we can understand how well that software works? And we're working at things like assurance cases and so forth to come up with scenarios under which we can evaluate equipment. And just as an example, simple things even aren't necessarily even bugs in the code, but user interface variations cause a lot of problems. So we have infusion pumps. You guys probably always use those. When you're programming infusion pumps and you move from vendor to vendor, you might have a different way when you punch up a number and you roll over the edge, where sometimes the number goes from 9 to 10, sometimes it goes from 9 back to 0. And those differences, when you may do that hundreds of times a day on different patients, cause medical errors to occur on the patients. And so seeing that you would think would be so simple and straightforward cause this sort of human interface interaction errors. And again, we're trying to work on ways that we can identify those errors and then come up with ways that maybe we could control those or make sure that they minimize the possibility of those types of errors. So it's a huge area. We don't have a lot of resources on how to solve all these problems, but it's really an area that's an important one that we're trying to deal with. So that's a real brief overview of what we do here. And I have a couple of individual projects. I won't go through them all. I'll just give you a quick summary. So what I try to do is just give you a pretty brief overview of the types of research we do in our laboratory. I'd say the other thing is we try to promote transparency and science-based decision-making. We do that through collaborative research, so we're very open to working with outside groups, other groups. We do a lot of work with universities, and industry as well. And then we make a lot of our tools available. We have a GitHub page where a lot of our in silico modeling is used by industry in their development of new tools, new imaging systems. We have a lot of analysis tools that companies and researchers use to do evaluation and to run studies. And we try to make that all available publicly for the use. So thank you.